All right, so today we're going to be talking about tissue culture. Um, so if we think about the human body, we have about 40 trillion total cells, and that's divided up into about 200 different types of cells across four major categories of tissues. And so, in order to figure out how any of these things work, one of the useful ways of doing that is to separate out each individual cell type and figure out how that cell type is working. We can also do the same thing for different disease states. We can model different diseases. And so if we get individual cells, we can figure out how things work. And so it should be noted that this wasn't a technique that was always available. Um, in 1953, cells were taken from Henrietta Lacks and grown by George Gay or George Guy, depending on your pronunciation. And he was able to establish out of these cervical cancer cells a stable cell line that have been known as the HeLa cells for Henrietta Lacks. It should be noted that these cells were taken without her permission. Um, Basically, it's one of the biggest human rights violations in science, but it's basically led to one of the great understandings of how humans work. And so there's a lot of debate around this topic, but we don't use HeLa cells in this lab. Um, but one of the things that was beneficial about these cells is that these cancer cells grow remarkably quickly, and all cancer cells are immortalized. They're they can basically be replicated for forever. And so there are ways to immortalize cells. So, like if you were to take your skin fibroblasts out of your arm and put them in a tissue culture dish, they would live for a little while and do some cell divisions, but eventually they would stop. The cells are not immortal; they can't continue on for forever. So, in order for cells to be really amenable in tissue situations, it helps if they're immortalized. And so, a lot of times we use cancer cells, but there are other mechanisms like forcing overexpression of telomerase that can lead to immortalization of cells. You can take cells right out of a person and grow them in a tissue. Well, a lot of cell types. Not every cell type is amenable to this. But if we take out certain types of cells and grow them for a short amount of time, those are called primary cultures as opposed to stable cell lines. Some of the cell lines that we use in the lab, we don't actually have any growing right now, but HEK 293 cells are ones that we commonly use. These stands for human embryonic kidney. We use them because they grow really quickly. They're relatively cheap. They were created from a legally aborted fetus in the Netherlands, and they were transformed with an adenovirus. And so, they're these cells, like stable cell lines that will grow for forever. Um, one of the weird things about them is that they don't really have much resemblance to kidney tissue, so they might not actually be kidney tissue. But they are an immortal cell line that is really easy to grow, and they're easy to genetically modify to learn things. The cells we're going to be using this semester are called NIH3T3 cells. These are mouse embryonic fibroblasts. They've been growing since 1962. And also, like the HEK cells, they are easy to get DNA into for genetic modification, and they also grow like weeds. And so these are what your cells are going to look like. So when we grow cells, one of the main things is the tissue culture media. Okay, and so we have a base media that has the core metabolites to support life. And so over here on the right, we have the list of things that goes up into tissue culture media. You can see there's a lot of amino acids, some vitamins, and some salts. One of the things that typically is added to the tissue culture media is phenol red, and this acts as a pH indicator. And so you can see it has this nice red color here. And so what you want to look for as you're culturing cells is that they should generally maintain this red color. Um, and so if the media gets more on the orange or yellow side, typically if it gets yellow, we're in trouble. Um, if it goes more towards the yellow side, that means that the media is more acidic. If it goes more purple, that means it's more basic. There are certain infections that can cause the media to change in either direction. Just normal metabolism of the media itself will cause the change from this red color towards the yellow color. And that's due to glycolysis secreting lactate from the cells, which acidifies the media. Um, and I should also mention that the base tissue culture media was originally designed by Harry Eagle in 1955. And so the base stuff to grow cells has been in existence for a while. Um, some other stuff that we do to add to make the cells grow. Typically, there's 5 to 10% fetal bovine serum. Okay, and so that's baby cow blood juice. So the serum is the non-cellular part of blood. And so that has a lot of growth factors in it, and that encourages the cells to grow. We'll typically also have antibiotics in the media, and that's just to prevent bacteria from growing in them. And so, like I said, as the cells grow, they'll metabolize the media. And so you need to change the media every 3 days. And so this is a really important thing. Once you get cells, they are your babies. I don't know if you ever had to do like the flour bag or the egg thing and in parenting in high school. This is even more important than those. Okay, so once you're allocated a dish of cells, you're going to be responsible for keeping that thing alive, or well, and propagating it, as we'll talk about in a second. And so at the very least, you need to come in every 3 days and keep track of your cells. If you don't, no eval. There are a few different media types that have been developed over the years. DMEM, or Dilbecco modified Eagle media, is sort of the main base media that a lot of cell types use. And so our NIH3T3s, the HEK293s, we have some colon cancer cells that we're actually growing in this now. There's also MEM alpha, which is typically for P19 stem cells that we use. DMEM slash F12K mix. This is also goes to certain colon cancer cells. This is something you'll see in a lot of immune cells. RPMI, which stands for Roswell Park Memorial Institute media, it was developed there. A lot of different immune cells utilize the RPMI. And so when you go to do tissue culture cells, tissue culture, if you look up your type of cell, you have to figure out what kind of media they require. And so this is actually sort of a problem because different labs tend to use different medias. And so you need to look through the literature and find the media that seems to be the most common. And one of the things that can happen is if a lab decides to diverge and they get their HEK293 cells grown in RPMI, those cells become acclimated to RPMI. And if you switch them back to DMEM, there can be problems. But it can also cause gene expression changes in the cells when you switch from one media type to the next. And so you have to be really careful about when you're doing experiments that you're using the right types of media. Also, the amount of fetal bovine serum that's present in the cells is also going to change gene expression. And so the combination of media type, the amount of FBS, we try to normalize for these things and keep them constant through experiments. Okay, so when we have cells growing in a dish, and these little blue sun things are my crude representation of cells, and this circle represents the dish that they're growing in. They proliferate and they attach to the dish. They should look all kind of spread out like we showed you in the pictures earlier. And eventually they're going to start crowding the entire surface of the dish. And we call that confluence. Okay, so if the cells are totally covering the surface of a dish, that's 100% confluence. They're totally confluent. Generally, we want to stop the cells from before they get to that stage. We typically want them to get to about 85% confluent. We always want a little bit of space in between our cells. And so letting them get over confluent or sort of growing on top of each other is a big problem. So never let your cells do that. So what we do is a process called trypsinization or cell splitting or passaging. And we add an enzyme trypsin to our cells. And so it's actually a combination of trypsin and EDTA that we add. And so trypsin is a digestive enzyme. And so what it does is it physically cuts the proteins that are on the surface of the cells that cause the cells to adhere to the dish. EDTA is an ion chelator, calcium and magnesium particularly. And a lot of the proteins that help cells adhere to dishes are dependent on calcium and magnesium. And so if you take away the calcium in particular, that causes those proteins to sort of lose their structure and it helps them fall off. And so the combination of trypsin and EDTA gets the cells up off the dish. And then what we do is we split them into multiple dishes. And so what I'm representing here is a 1-to-3 split. So we've taken all the cells off of this dish and we've divided them into 3 different dishes. This splitting ratio is going to be dependent on the type of cells that you're working with. Some cells require a low split. So like 1-to-3 is considered a low splitting ratio. 1-to-6 is a fairly common splitting ratio. So if we had taken all these and divided it up into 6 dishes, that would be a 1-to-6 ratio. And sometimes it goes down to like 1-to-10, 1-to-15 if the cells are really fast growing. So you add your trypsin, put the cells back in the incubator. The trypsin is active at 37 degrees, and so that encourages the cells to come off. Take them out of the incubator. We inactivate the trypsin with tissue culture media. The tissue culture media has trypsin inhibitors in it. If you leave your cells in trypsin too long, the trypsin will actually digest your cells. And so you always have to add media to inactivate the trypsin when you're done. Once you do that, you get your cells all nice and evenly resuspended by pipetting, and then you're going to split the cell suspension into multiple dishes. Sometimes you might not actually want to make multiple dishes. Say that your cells are getting confluent, but you don't have any experiments planned for them. You still have to split them. You need to give them room to grow. And so, if in this example, if we only did one dish, but we used 1/3 of the cells, that's still a 1-to-3 splitting ratio. And that tends to be the hardest part for students to grasp. Okay, so if we're doing a 1-to-6 split, it would be, taking this number of cells, dividing it into 6. Even though we're only making 1 or 2 dishes, you can still do a 1-to-6 split. Okay, and so just think about that splitting ratio. It's just a fraction of the total cells that go into the new dish. Okay. Once you put the appropriate relative volume into a new dish, you have to think about the volume of media that should be on there in total. And so say that you're only putting 0.5 mils of your cell suspension onto a new dish. You can't leave just 0.5 mils of cell suspension on that dish because it'll evaporate. You need more volume to sort of make the cells happy and cover the surface of the dish. And so 35 millimeter dishes are the dishes that we use most commonly. The total volume of media should be 1.5. You can get away with with 2 mils of media, but it gets harder to deal with. And so 1.5 is our target volume for a 35 millimeter dish. 60 millimeter dish tend to have 4 mils of media on there. T75 flasks, you'll see some of these in the incubator, but you won't be working with them this semester. They have a total volume of about 15 mils. If we do things like 96 well plates, so if you remember the first lab of this semester when we did the 96 well plates, those only use like 1 to 200 microliters of media total. And so you have to think about like the surface area of your dish, how much media you want to use for it, and account for that. Whenever you split your cells, you always want to label the type of cells, right? So if we're doing NIH3T3 cells, that's the top thing that goes onto your dish. You want to include the splitting ratio. So the last time you split it, what was the splitting ratio? Like 1 to 3, 1 to 6, whatever. The date, the passage number, and your initials. Okay. And so the passage number refers back to how many times these cells have been split. And so when we get them, a lot of the times we get our cells from an organization called the American Type Culture Collection, which is a huge repository of cells. When we get the cells from them, we consider that passage 0. The cells haven't been split at all. Then the first time we grow them and split them, that's their first passaging. And so we call that passage 1. And then when this one grows up and we split it, we call that passage 2. And we keep track of that. You generally don't want to do more than maybe 25 to 30 passages on a cell line because it tends to diverge from its original cell type, because you're doing a selective pressure by growing the cells in a tissue culture dish versus cells in their normal environment. Um, so we try not to passage our cells too many times. So what are we going to do with our cells once we split them? Well, so far this semester, you've been working on cloning a couple of genes for Amy. You're going to be taking those once they're in their plasmids and transfecting those plasmids into the cells. And so we're going to see what Amy's proteins look like once they're expressed inside the cells. And so transfection is the general term for putting nucleic acid. It can also technically be protein, but in general, it's more common to transfect nucleic acid into cells. And specifically, transfection is putting nucleic acid into eukaryotic cells, typically derived from mammalian or higher uh, multicellular organisms. Um, trans Transformation, if you remember from a couple of weeks ago, it's putting nucleic acid into prokaryotic cells. So like taking DNA and put it into bacteria. Um, Transformation is also taking DNA and putting it into yeast cells, so single-celled organisms. Um, and so it's interesting, the transfection that we're doing is a liposomal transfection. So basically, we add some reagent with our plasmid vector, we kind of mix the two together, let them hang out for a little bit. They form this DNA lipid complex. We add that to the cells. The cells take up that DNA, and somehow it magically goes into the nucleus and starts being expressed. It's kind of interesting to note that we still don't quite understand how the DNA makes its way from the cytoplasm into the nucleus, but let's ignore that. Um, I also wanted to point out that the Pfizer and Moderna vaccines, if you're going to be getting a vaccine, they use a little piece of the viral genome in a lipid carrier to transfect a little chunk of the viral genome and express that into our cells. And that expression of the viral protein is what causes the immune system to mount a response. There's also a mechanism where we can use viruses to deliver nucleic acid. And so there's an example of a adenovirus or adenovirus, depending on your pronunciation preference. This is called transduction. It should be noted that the Johnson and Johnson vaccine does a similar thing where it tries to it expresses a little bit of the viral genome in our cells, but that vaccine uses adenovirus to deliver the genome, and actually delivers DNA. That DNA is decoded into RNA, and then the RNA is encoded into protein. The Pfizer and Moderna ones are actually delivering RNA directly into the cell. This is a much more efficient process, and that's probably why the Johnson and Johnson vaccine isn't quite as effective as the other two. Um, anyway, back to what we're doing. So we're going to be putting our plasmids into our cells. We're just using regular old transfection with a lipid-based reagent. And so once our plasmid gets inside the cell and makes its way into the nucleus, this CMV promoter stands for cytomegalovirus. This promoter is very active in mammalian cells, and so it starts expressing whatever's behind it. If we haven't inserted a gene here, then we get expression of our GFP, or if you use the cherry plasmid, you're getting expression of cherry. But hopefully you have your gene of interest here, and you're going to get a fusion protein of that gene of interest in combination with the GFP. We're going to be doing a process called transient transfection, where we add the DNA to the cells and we use them 48 to 72 hours later. But theoretically, you can also make stable cell lines. And so we've mentioned before that there's an antibiotic resistance cassette here. And this provided our kanamycin resistance in the bacteria, but this one actually has a dual promoter, which is kind of fun. And so this promoter is also, or it's actually two promoters right after each other. This promoter can be active in the mammalian cells. And so if we add the antibiotic G418 to the cells, it'll kill cells that have not, that are not expressing this plasmid, or that do not have the plasmid in them. And so theoretically, we could make stable cell lines that all of the cells, like we do with the bacteria, all of the cells would have our plasmid, and so we could do more long-term experiments with our genes of interest. But for our purposes, we're only doing transients. So I wanted to explain the concept of fluorescence. I know I've brought up green fluorescent protein several times, but let's talk specifically about what happens during fluorescence. Basically, it's a property of a compound to absorb light at a particular wavelength and emit light at a different wavelength. And this always has to happen where it absorbs light at a shorter wavelength and it emits light at a longer wavelength. Okay, and so that's due to physics. Shorter wavelengths have higher energy, and so the higher energy light comes in, bounces around the compound, excites the compound, and then as the compound goes back to its lower energy state, it releases a photon light that is at a longer wavelength because it has less energy. So the compounds that have these properties are called fluorophores, and this is an example of a fluorophore called fluorescein. And so fluorescein absorbs in the blue or ultraviolet wavelengths, and it emits light in a green wavelength. And so you can see it here. And so every fluorescent compound is going to have an ideal wavelength that it absorbs light, and that's what's shown here. And then it's going to have the wavelengths of light that it emits light. And so you can see that there's some overlap between the excitation wavelengths and the emission wavelengths. But overall, we can apply filters and stuff and measure specific wavelengths here. Okay. So, the GFP allows us to do some fun stuff. And so, most easily we could see where it goes around inside the cell, localization, but also we can use it if there are not good antibodies against a protein of interest. We can, detect the protein of interest through the GFP, because there's lots of good antibodies for GFP. And so we can see our protein in other assays. But we can also use the GFP as sort of a way to purify proteins of interest. So if our protein is attached to this GFP and we pull down the GFP, then our protein is going to come down. So there's lots of different things you can use. And it should be noted that GFP was originally purified out of a jellyfish. It was part of a dual protein complex where this corn, um, and once it gets excited, emits blue light, and the GFP absorbs the blue light and emits green light. Um, Over time, GFP has been genetically modified. So, technically, our plasmid name is EGFP for enhanced green fluorescent protein. Our green fluorescent protein has some mutations in it that enhance its spectral properties, and clean it up a little bit. Um, and so, you know, we're using a variant of EGFP, but there's lots of you also doing the M cherry, which is a modification of a slightly different protein, but essentially we have a full array of fluorescent fusion proteins that we can use nowadays. Um, so how do we do this when it comes to the microscopy? And so if we think about regular light microscopy, and fluorescent microscopy is technically light microscopy, but the kind of microscopy that you might be more familiar with is called phase contrast microscopy. And in this technique, we have a light source, and this is a regular type of microscope. This whole process can be flipped over in what's called an inverted microscope, and our tissue culture microscope is actually an inverted one, but just think of this whole thing as upside down, compared to what I'm telling you here. But so we have this light source down here. The light source comes through. It goes through this thing called a condenser annulus, and then it comes up through here, and our sample would be right here. And what the condenser annulus does is it sort of bends light out in a weird direction. And so as that light comes up, it's going to pass through our sample in kind of a weird direction. And so then on the other side, we have our objective. Our objective is the thing that actually does the magnification. But on the other side of the objective, we have this thing called the phase plate. And what that does is it takes that weird refracted light and it bends it back, and to help refocus it. But in the combination of the between the condenser and the phase plate, we're adding contrast to our sample. And so if you look here, if we were to look at the cells without the phase contrast microscope, you can see they're like basically clear. Okay, so what we're seeing is sort of a shadow of the cells here. Okay, so we're seeing the cells, whatever light kind of gets blocked by our sample is shows up as different intensities. And so cells are really thin things, and most of the time they don't have a whole lot of stuff that's going to diffract light and allow you to see a shadow. So what the phase contrast does is in that combination of breaking it apart or pulling it out and bringing it back together, it adds contrast. And so this is the same image in open bright field versus phase contrast, and you can see all the contrast that's been added. Some of the problem with it is you get like these halo effects around your samples, but it allows us to see our samples clear. But in both situations, again, our cells are on a thing, the light passes through them, and we're seeing a shadow of essentially the light that got stopped by our cells, which is not a ton for normal tissue culture stuff. So with fluorescence microscopy, we can do something very different. And so with fluorescence microscopy, we have a light source that comes in, and we have an excitation filter. So if you remember with a fluorophore, we have a specific excitation and emission wavelength. And so the excitation filter will only allow certain wavelengths of light to move through. And so here in this example, we have blue light coming through our excitation filter that's going through the objective and hitting our specimen. And so our specimen here has a green fluorophore on it. So that blue light excites the fluorophores. That blue light exciting the green fluorophore causes the fluorophore to emit green light. And then we have this dichroic mirror that bounces the light down and then allows this light to pass through. And again, our objective is the thing that actually doing the most of the magnification here. And so the emitted light's going to come through here, and then it's going to pass through an emission filter. And so this is only going to let green light go through, because even though we're moving the light in this way, we're still going to get some blue light that's going to want to come back up through here. And so we're going to add an emission filter so that like only the green light is coming through. And so as opposed to phase contrast, where we're looking at sort of a shadow of our sample, the fluorescence microscopy is you're actually looking at the light that's coming directly out of your sample. And again, these can be inverted, so it doesn't necessarily have to look like this. All of this stuff could be on the bottom versus on the top. But again, shadow, light that's actually coming up off of our sample. Okay, so what we're going to be doing is transfecting the cells and doing fluorescence microscopy on them. And so, I sort of mentioned this with the variations of GFPs that are available. We have other fluorophores available, so we can do multiple stainings of the cells at a given time. And so if we add different stains to our cells and they all have different spectral properties, then we can use different excitation and emission filters. And so one of the dyes we're going to use, it's an ultraviolet excitation filter. So it's going to let ultraviolet light onto our sample. Our sample is going to be emitting blue light. We're going to have one where we're exciting our sample with blue light. We're going to see the green light come off. Then we're going to have one where we excite our sample with green light and see the red light come up off of that. Okay, and so we have a lot of different fluorophores. So ones that we're going to be using in this class, or stains that we're going to be using in this class. The stain is called phalloidin. This is a toxin that comes from the death cap mushroom. And what this does is it binds between actin subunits in the actin cytoskeleton. So like where my thumb is, it basically gets in there between the actin subunits, and it locks the actin subunits together. And basically it's a way for us to stain the actin cytoskeleton inside of cells, because it binds inside there. And so people have taken that toxin and they've added fluorophores to it. And so we purchase it. So we have phalloidin that has a Alexaflor 488 or Alexaflor 555 fluorophore that's been covalently attached to it. So wherever the phalloidin binds, the fluorophore is also going to be there. And so the 488 is a green and the 555 is a red. And so if you're transfecting your cells with green fluorescent protein, you want to use the 555 or the red phalloidin. If you're transfecting your cells with the cherry, you want to use the green phalloidin, because we want two different spectral properties, right? We want your gene of interest, but we also want to see the phalloidin, but they need to be two different colors. And so this is something that you really need to think about every time you do a staining. Think about all the different colors that you could potentially use. So make sure that you're doing a different color than what your plasmid is. Okay, the other one that we're going to do is DAPI. That's the full name for it. This one binds to DNA, and when it does, it has enhanced fluorescence, kind of like ethidium bromide. But this one gets excited by ultraviolet light, and it emits blue light. And so wherever your DAPI is, that's where the bulk of your DNA is. And you can see the nuclei very nice and clearly when we stain our cells with DAPI. Okay, so this week, what you're going to do, you got to make sure that you have your plasmid DNA. You need your plasmid DNA with your gene of interest, and then you'll also separately need your plasmid DNA that doesn't have an insert in it. You're going to need a dish of cells, and you're going to split that dish into multiple dishes. I want all of you to, this is definitely a technique that you can't do sight unseen. You need to schedule it specifically either with me or as a TA. And this isn't something that we can do with a lot of people at one time. It's basically one or two people um, per session. And so you're going to get a dish of cells and you're going to split that dish into multiple dishes. Then the next day, you're going to transfect your dishes with your clone gene of interest or the empty plasmid. Um, and so, yeah, whatever you're going to do. So make sure that like if you cloned into the PM Cherry N1 plasmid, make sure that you have some PM Cherry N1 without any insert in it so that you can do both of them. And so like in your group, if one person wound up doing Cherry and one person did GFP, then you're each going to have one Cherry with your insert, one Cherry with your not, with no insert, one GFP with your insert, one with not. So you're going to do the transfection 48 hour or 24 hours after transfection. We can look in the microscope and see if the cells are starting to show any color. But also at this point, you need to feed your cells. At 20, after the transfection, we want to get the residual transfection reagent off and give them new food. But then sometime between 48 and 72 hours post transfection, you're going to fix and stain your cells with phalloidin using the opposite wavelength of your transfection for both, all of your dishes if you're working in a group. Okay, and that's it. We're going to take pictures of them. It's going to be super pretty, and hopefully this will be a nice little reward for all of the work that you've put in because these pictures are wicked cool. All right. Talk to you soon.